So we've been talking about tubular reabsorption, sodium, glucose, bicarbonate, potassium. We've talked to some about importance of water reabsorption, especially in the proximal tubule where water reabsorption accompanies sodium and glucose reabsorption. Now we're going to focus on the loop of Renal. And we're going to talk about control mechanisms for water. We're going to focus on water homeostasis. It's an important part of what the kidney does, controlling the excretion or retention of water as needed by the body. So here's some typical values. You typically drink about 1.2 liters of water. You get about 1 liter of water from your food, the moisture in your food. You produce about 300 mils of water. Do you know where you produce what? You produce water. Do you know where you produce water? Metabolism. Why do you produce water in metabolism? Well, it says it right there. Why do you produce water in metabolism?
It's a byproduct of the citric acid cycle?
Nope, not of the citric acid cycle, but you're really close. It is the final product when oxygen is the final electron acceptor of the electron transport chain. Oxygen is oxidation is loss. Oxygen is reduced to water. So you produce water in the process of creating ATP by oxidative phosphorylation. There are some animals, by the way, desert animals, that get none of their water from drinking. They don't drink. They only get water from the seeds, the dry little seeds that they eat, and the water that they get from metabolism. That's their source of water. It's incredible. Anyway, this is us. This is our sources of water, and here are our excretions of water. We urinate about 1.5 liters a day. We produce about 100 mils of water in feces. We sweat away about 500 mils of water, and we exhale water vapor about 3 to 4 liters, 3 to 400 milliliters of water per day. So this is water homeostasis. A few years ago, I did an experiment with students like you in A&P, and I measured their urine osmolarity as a function of time. And so the osmolarity, remember, is just the solute concentration, total solute concentration. Sodium, potassium, urea, presumably not glucose, because hopefully none of these students were diabetic. But those are the main solutes in urine, and osmolarity is just the concentration of all of them together. So you can see a wide variety of students walking into the lab. That is to say, some of them were fairly dehydrated and were producing a urine that was very concentrated, almost as concentrated as possible. And some of them were walking into lab producing a fairly dilute urine that was actually more dilute than the body osmolarity, which is about 290 milliosmoles. So, just a little bit hypotonic, below the osmolarity of normal body concentration. And so obviously this person was relatively water deprived, and this person had been sipping on a bottle of water, all morning or something, and in between. So a wide variety of urine osmolarities depending on the drinking of the person. And then 500 mil, at time zero, they all drank a half a liter of water, just straight up water. And their osmolarity all fell to, either isotonic or hypotonic, depending on where they started. Tonic. So when I say hypotonic, that means the osmolarity is below 290 milliosmolar. And hypertonic is above 290 milliosmolar. And isotonic is 290 milliosmolar. And here's a different group of students. Obviously, again, some relatively dehydrated and producing a concentrated urine, and some relatively hydrated, quite hydrated, and producing a dilute urine. And then they were water deprived for an hour or two. And so the ones that were dehydrated stayed dehydrated, as you would expect. And the one that came in fully hydrated, the osmolarity of their urine rose within an hour and then stayed relatively high. So this is evidence from people like you from not too many years ago of the variation, individual variation, and individual responses to drinking water or not drinking water. So now we want to talk about the mechanisms for water homeostasis. So here are the themes. These are the themes that we're going to be elaborating on for the course of the day, or the hour. The kidney generates a concentrated urine as needed if a person is relatively dehydrated and wants to conserve water, a concentrated urine, by using the loop of Henle to build a hypertonic renal interstitium, a hypertonic peritubular space. The loop of Henle is going to build a hypertonic peritubular space. Then osmosis is going to pull water from the urine, from the tubular fluid, into this hypertonic peritubular space through water permeable channels, we'll see those, and then recover it into the vasa-recta capillaries. So we're going to build a hypertonic interstitium and then use osmosis to pull water out of the urine and recycle it back into the blood where it goes back to the body. And it is the collecting tubule where this water permeability is going to be most important. So the loop of Henle is going to build the hypertonic peritubular space, and the collecting tubule is then going to allow the water to go into that hypertonic peritubular space and then into the vasa-recta capillaries. Okay, so that's where we're going. So the idea is that tubular fluid is isotonic in the proximal tubule because we absorb a bunch of salt, sodium and chloride. We absorb a bunch of sodium, but we also absorb water. So what we absorb is isotonic, and the fluid stays isotonic because it started isotonic. It was just a filtrate of plasma. So by definition, it was isotonic, and it stays isotonic in the proximal tubule. The fluid in the tubule is going to become hypotonic by the end of the loop of Henle, because that's the part where the loop of Henle made the hypertonic extracellular peritubular space. So if the extracellular peritubular space is becoming hypertonic, the fluid is going to become hypotonic. And then in the collecting tubule, the collecting tubule has mechanisms for either excreting a hypotonic urine, in the case of a hydrated person, or a hypertonic urine, in the case of a dehydrated person, in order to control the concentration of solute in the fluid and decide whether water excretion or water retention is necessary. Remember that you can die of dehydration, you know that, and you can die of hyperhydration. We have to keep it within a range. People have died of drinking too much water. Okay, and vasopressin. So vasopressin is a hormone that is going to become really important in this control. Okay, so we're focusing on the loop of Henle, the vasa recta, well, let me do this right. The loop of Henle, the collecting tubule, and the vasa recta. And obviously this is the cartoon you're used to seeing, but you know from lab that this is all splayed out, and this space, this peritubular space, is extremely exaggerated. All of these things are clumped together very tightly. You saw that in lab. So that's the image I want you to have in your mind of this tube being right up next to this tube, being right up next to this tube, with these capillaries squeezed in there as well as the peritubular space squeezed in there. Remember those images from lab. Okay, so now we're starting to tell the story of the loop of Henle and how it creates this hypertonic peritubular space. Okay, this slide gives everybody a headache, not just you, it gives us all a headache. But let's walk through it. Remember that fluid is flowing from the proximal tubule into the descending limb, and then it goes up the ascending limb into the distal tubule, and then down in the collecting tubule. So that's the flow of urine, or tubular fluid. Input here, output here. The key thing to know about the descending limb is that it is water permeability. It is that it has high water permeability. And it does not have a lot of solute transport mechanisms. So in the descending limb, we need to know that there's water permeability. Stay with me. We'll figure this out. As you can tell, I'm trying to make it simple, but it's complicated. So I'm walking you through it in a way that I hope will make sense by the end of the story. But it's complicated. I know that. Water permeability in the descending limb. Low water permeability in the ascending limb, and high sodium and chloride transport. So now, let's not worry about urea yet. So this arrow here that is showing sodium, let's focus on sodium, pumping. Remember, there are two membranes here, right? There's the apical membrane and the basolateral membrane. This line is an epithelium, and so this line has an apical membrane and a basolateral membrane. What is the primary way that sodium and chloride get through the apical membrane of the ascending limb of the loop of Henle? What is the name of the transporter?
NAK2Cl.
NAK2Cl co-transporter in the apical membrane of the ascending limb. And what is the way that sodium gets out of the basolateral membrane in the ascending limb? The sodium potassium ATPase. So these active transporters, these single arrows, are really two serial transporters. The sodium potassium 2-chloride co-transporter and the sodium potassium ATPase. Okay? But the big picture is, and we have a lot of expression of those two transporters in the apical and the basolateral membrane, and so we pump sodium, chloride follows, let's not worry about potassium. Let's focus on sodium here. So sodium is being pumped out of the tubular fluid as it goes up the ascending limb. So the fluid that enters the distal tubule is hypotonic because that solute has been pumped out of it. And there we see these numbers. Remember, isotonic is 290, which in this slide is rounded to 300. But here it's hypotonic. And it stays hypotonic until it reaches the collecting tubule. Here it shows it becoming isotonic by the end of the distal tubule. Let's not worry about that. It stays hypotonic until it reaches the collecting tubule. And then, in the collecting tubule, we're going to have a variable water permeability, determined by vasopressin. A vasopressin-dependent water permeability. If we need to produce a concentrated urine because of dehydration, vasopressin, which will be released in response to a dehydration signal, will increase the water permeability of this tubule and allow water to flow out of the fluid into the hypertonic interstitium, which I have not yet completely described to you, and produce a concentrated urine. If we are highly hydrated and we need to produce a dilute urine, vasopressin will be absent, water permeability will be low, and the dilute fluid that is produced here will be excreted. Okay, that's where we're going. Now, let's see, what is this? So the loop, like I said, this process of water permeability and sodium chloride active transport in the descending and the ascending limbs respectively, and where it happens is important, creates this hypertonic interstitium and the hypotonic fluid leaving it, and then the collecting tubule has variable water permeability, either to produce a highly concentrated urine, which is illustrated here, or a dilute urine, which is not really illustrated in this particular slide. Okay, so that's where we're going. Now I want to describe how this loop-de-loop thing, the loop of Henle, the descending limb with water permeability and the ascending limb with salt transport, how this geometry and pumping process creates a hypertonic interstitium. That's the magic right now that you may not fully understand. And by hypertonic interstitium, I want to just say that the deeper we are, so remember this is the cortex up here, which is generally isotonic, 300 millimole. Here is the loop of Henle going into the medulla. And when I say a hypertonic interstitium, I want to point out that the interstitium is most hypertonic, deeper in the medulla, and is less hypertonic, shallow in the medulla. The closer you are to the cortex, the closer you are to isotonic. The deeper you are in the medulla, the more hypertonic the peritubular space is. It's a gradient, a solute gradient. Low solute concentration in the peritubular space at the corticomedullary junction, and more concentrated as you go deeper in the medulla. That's what I'm trying to tell you how do we do that is what's coming next. Okay, yes?
So, the further you are in the medulla, the more hypotonic?
The deeper you are in the medulla, the more hypertonic you are in the peritubular space. Okay, so let's see where this comes from. Okay? It seems magical, I think, but here's how it works. So let's just start with a loop of Henle that has no gradient. It's all isotonic. Okay? That's the simple. Let's just start there with an isotonic loop of Henle. It never exists, but that's where we're going to start in trying to build this story. Okay? This is where we start. Isotonic everywhere. That's what all those 300s mean. Let's turn on the pumps in the ascending limb of the loop of Henle and turn on the water permeability of the descending limb. So, and remember, these pumps are two pumps. It's the sodium-potassium 2-chloride cotransporter in the apical membrane, and it's the sodium-potassium pump in the basolateral membrane, right? Both of those processes are embedded in these arrows and pumpers. That's what this is. So we pump solute. It started at 300, we pump solute out of it from here to here. So this concentration goes down, and this concentration goes up. It goes up, and so it goes up because we pump solute into it, and so the concentration of this goes up because water moves from here to here to equilibrate. High water permeability means that there's going to be an equilibration in the descending limb of the solute concentration. So the pump makes a hypotonic fluid, makes a hypertonic peritubular space, and then equilibration makes a hypertonic descending fluid. Okay? So far, everything seemed believable, right? I'm not going to get all the way there, but I'm going to make it believable. That's what I want you to believe it. Okay? So do you all believe that this situation happens after the processes that I've just described? Hopefully you believe it. Now we're going to introduce a little bit of new isotonic fluid. There's flow, right? So these two 300s were introduced into this tube, right? That's the shift. We're going to push in to these two 300s. They came from the proximal tubule. Okay, so everything shifts. So these 300s are new, these 400s used to be these 400s used to be these 400s, these 200s used to be these 200s, and these 200s are now out in the distal tubule. Okay, we've moved two numbers. But that's fluid flow, right? And so we've shifted it, and now this is the situation we have. We have not yet, so why are these 300? Because these 300 have equilibrated with the water permeability of these new 300s. And these 400s have equilibrated with this, and we haven't even had to do any more pumping. All we've had to do is to shift it and then allow equilibration of water on the descending limb, water permeability. Because this is so water permeable, this concentration is set at this concentration, and this concentration is set at this concentration. Water permeability does that. Well, you can see already we have started to build a gradient. Just what we've done, we've started to build a gradient. It is isotonic in the shallow medulla, and it is hypertonic in the deep medulla. Not very, but there is the start. So let's continue to do this. Let's do one more step of pumping. We're going to pump from this 200 into this 300, so it's going to be 150 and 350. We're going to pump from this 200 into this 300. It's going to go from 150 to 350. We're going to pump from this 400 to this 400, so it's going to go to 300 and 500. These numbers are more or less made up, but you can see what is happening. We're pumping solute out of this space into this space, creating higher concentration here, creating lower concentration here, and then equilibrating, as we always do, here. Okay? Now we have even more of a gradient. And if we shift and equilibrate, so here's two new 300s. Here are the 500s that were, let's see, how does this work? Here's two new 300s. Here are 500s that came from these 350s. These 500s came from these 500s. So let's not worry about the numbers too much. The point is, we're bringing in two new 300s, and then we're equilibrating across the descending limb. Well, here's a real gradient. Here is a dilute exiting fluid. Do this a few thousand of times, and remember it's a continuous process. It's not just, we're breaking it down into pumping and equilibrating and shifting and equilibrating, but it's a continuous process. Fluid is moving slowly, and these things are happening continuously. And by these things happening continuously, we build a gradient, something like this, if we are dehydrated. Believable? More or less. There's the gradient we're talking about. Isotonic at the corticomedullary junction, hypertonic deep in the medulla. That's the building of the peritubular gradient. We haven't used it yet in the collecting tubule, but we've built it. It's called the countercurrent multiplier. It's called a countercurrent multiplier because it depends on these two things being in close contact with each other. I can get into the biophysics of countercurrent multipliers in a number of systems, but let's not do that. This loop is called a countercurrent multiplier. It's multiplying the gradient. Linear systems don't do this. It depends on the geometry. And you can predict. So those desert rats that drink no water and get all their water from metabolism and from a little bit of moisture in the seeds that they eat, what do you think do you think their loops of Henle are longer or shallower than ours? Longer. They have a much longer, a much deeper loop of Henle, and they can create a solute gradient going from isotonic, they're mammals, so they have an isotonic saline much like ours, and deep in their medulla it's probably 3 molar sodium chloride. It's almost crystalline, it almost crystallizes out. I mean, it's almost saturated. And so they produce a urine that has almost no water in it. I mean, it's obviously they excrete some water, but not very much. They conserve water because they don't drink any of it. Water rats, so rats that live in swamps and stuff. What do you think their loops of Henle look like? Pretty shallow, because they're constantly trying to get rid of excess water. They don't need to, they never have to live in an environment where they have to conserve water. So they don't need deep loops of Henle. We're somewhere in between, a water rat and a desert rat, in terms of our kidneys. The vasa recta, so here's another slide that gives you a headache. The vasa recta parallels the loop of Henle. If this loop of Henle were perfused by blood, if this loop of Henle were perfused by blood that was isotonic, so if we had blood flow across here that was isotonic, right? I mean, we need blood, both to provide the loops of Henle cells with the metabolism, glucose and things they need to make ATP, because they're burning a lot of ATP pumping all that solute. So they need blood, and plus they need the blood flow to recover the water that they're trying to recover from the collecting tubule, which we haven't got to. But if we had blood flow that looked like this solute gradient would get washed out, because we had 300 milliosmoles flowing in, and we have water permeability here, and so we would be washing out the gradient that we've worked so hard to build. But we don't do that, because the vasa recta doesn't, the blood flow parallels the loop of Henle flow. And so what flows out is only slightly hypertonic to what flowed in, instead of being dramatically hypertonic. Is that clear? So the loop of Henle creates the hypertonic peritubular space, and then the vasa recta provides blood flow and preserves the hypertonic peritubular space. All these arrows I don't even know how to interpret this. It's from your book. I couldn't find a better one. So there's no good figure that's showing. It's showing water flow, water flow from water flow is going from dilute to concentrated. Let's not worry about the arrows and the numbers. The point is the vasa recta preserves the solute gradient better than if we just had, a mishmash, a peritubular network of, peritubular capillaries that is just a meshwork like we have up in the cortex. Okay, so now we have to use this gradient that we worked so hard, that your loops of Henle burned so much ATP to produce. The main point of this figure is that the proximal tubule has a high water permeability. There it is. Proximal tubule, we're forgetting about convoluted and straight. The proximal tubule has a high water permeability. You already know that. This is data that produced that. These are actual data. The descending limb of the loop of Henle, let's not worry about thick and thin, the descending limb of the loop of Henle has a high water permeability, which I just told you about. That was part of the countercurrent multiplier. High water permeability in the descending limb, making for equilibration in the descending fluid. Okay, so both of those things you already know, and now I'm proving it. Other parts, especially the distal tubule and especially the distal tubule, have low water permeability. So the hypotonic fluid leaving the loop of Henle stays hypotonic in the distal tubule. But here, now we're getting to the interesting thing. Well, this has all been pretty interesting. Now we're getting to the payoff. And that is in the collecting tubule, we have variable water permeability depending on vasopressin. So we have vasopressin dependent water permeability. If you are dehydrated, you produce vasopressin. The vasopressin tells your collecting tubule to make water permeability so that, well, let's go to the next slide. Let's go to the next slide. Okay, so this is the second of these two slides. If you are dehydrated, vasopressin tells the collecting tubule, make water permeability, and so water is now abstracted or sucked out of the tubular fluid because of this hypertonic interstitium. If you are not dehydrated, I just went backwards. If you're not dehydrated, you don't make as much of a hypertonic gradient, and you don't use it because you're producing a dilute urine. Okay, so let's use this slide. Let's go forward in time, and then let's use this slide as just to remind you of what's going on, sort of big picture. Isotonic coming in, hypotonic going out. We're excreting water. This is someone who has just drunk a liter of water and needs to excrete water. Isotonic going in, hypotonic coming out. Water permeability in the descending limb, sodium pumping in the ascending limb. You know about that. Creating a hypertonic, but not very hypertonic, interstitium, peritubular space. The vasa recta preserves it. The collecting tubule ignores it and produces a dilute urine. Okay, now let's go to this slightly more interesting case of dehydration. But the story is the same with just two variations. One is and I'll explain them both at the molecular level, as you know I love to do. Same general principle, right? Water permeability, pumping, producing a hypertonic peritubular space deep in the medulla. Why is this more? The answer is we use a hormone to tell the ascending limb, make more sodium potassium ATPase and make more sodium potassium 2-chloride cotransporters. Turn this on. By turning this on and leaving everything else the same, we make a more effective countercurrent multiplier. And so it makes a higher gradient in the deep medullary interstitium. So a hormone, which I'll get to, even though this isn't endocrinology, I'll talk about the hormone. More solute pumping, loop-de-loop biz, loop-de-loop preserve, the vasorecta preserves the gradient that the loop of Henle built. And now the other interesting thing is a hormone of vasopressin-dependent water permeability to use this gradient, pull water out of this tube, and excrete a concentrated urine in order to preserve water, because we're recovering water here. Here, I'm going backwards, here we're excreting water, and here we're recovering water due to vasopressin-dependent water permeability in the medullary and cortical collecting tubule. Yeah?
So when we want to hold on to water, we ensure that the concentration of NaCl is higher in the peritubular state.
That's right. We ensure that the concentration of sodium chloride, and it turns out urea, is higher in the peritubular space, and we make sure we have a mechanism for using that concentration to pull water out of the urinary fluid by osmosis. Those are the two things. Build a concentrated peritubular space, and then use it to pull water out of urine by water permeability. So how do we do that? Let's talk about the water permeability first. Now we're getting into molecular mechanisms. But you know all this. This looks complicated, but you already know all of this from cell-phys and from A&P. What is this? You're going to tell me the story. I'm going to point to it. What is this? Let's get oriented. Here's the tubular lumen. Here is the basolateral membrane. This is a medullary collecting tubule epithelial cell. An epithelial cell in the medulla collecting tubule. What is this? Well, here it is. It's given to you. Here's the receptor for vasopressin. I've told you it was a vasopressin effect, so you already knew. Okay, there's got to be a receptor for vasopressin. If you didn't know that, you could have figured it out with your head. Receptor to vasopressin. What is this? GS. What is now going on in your brain? Not shown there. Somebody tell me something that's going on in your brain that is relevant. Okay, well, that's shown there. Well, I'm thinking before we get to activation of AC. Conformational change, that's in your head. What else is in your head?
Alpha subunit.
Alpha subunit. What's happening to the alpha subunit? GDP coming off, GTP coming on, more conformational change. Separation of the alpha from the beta gamma subunits. Right? It's all there. It's all there. Alpha turns on AC. What is AC? Adenyl cyclics. What does that do? Make cyclic AMP from ATP. Let's not worry about this inhibition here. So we're turning on adenylyl cyclase. We're making cyclic AMP. What does cyclic AMP do? It binds to the regulatory subunits and it exposes or it relieves, it allows the catalytic subunits of pKa to do their thing. Phosphorylation. Now we're getting into cell phys. No, we're getting into cell phys. We're going back to cell phys. If cyclic AMP can phosphorylate a transcription factor called Krebs and tell it to make protein, to increase transcription. That's one thing it can do. And the thing that it makes, that it transcribes, is coming up. We don't even have to go to transcriptional control though, because pKa can tell the vesicles that are containing, what is this supposed to illustrate? Aquaporin, a water channel. This water channel is existing in these pre-secretory vesicles. Phosphorylation of them, remember we've got constitutive and controlled exocytosis. We have some controlled exocytosis going on here. We take this aquaporin channel in these pre-exocytotic vesicles, and we put them in the apical membrane. So now we have water permeability, and we have constitutive water permeability here, more aquaporins. And so we now have made a vasopressin-dependent increase in water permeability in the collecting tubule epithelial cell. Vasopressin receptor is a GS-coupled receptor. You're often running on the GS story, pKa, and now two of the substrates that I want you to remember from self is Krebs, the transcription factor, and aquaporin, the channel, which then gets inserted into the apical membrane to increase the water permeability. So in the absence of vasopressin, these vesicles are endocytosed, and you remember endocytosis, clathrin, dynamin, targets, you remember all that. It's in there. You just got to pull it out in the right time, in the right place. Clathrin-coated pits, not shown here, but that's how these vesicles would get endocytosed, recovering, decreasing the water permeability in the apical membrane. So you drink a ton of water, vasopressin goes down, this system gets shut off, endocytosis gets turned on, and we decrease the water permeability, we produce a dilute urine. You stop drinking for 12 hours, you become dehydrated, your brain says, I'm dehydrated, makes vasopressin. Vasopressin does this, increases water permeability, and you pull water out of urine more and recover it better. Piece of cake, right? No, it's not. I do know it's not a piece of cake. I know that it's hard. But if you tell yourself the story, if you outline the story, if you take a slide, maybe one trick here would be to take some of these slides and white out or blank out or just cross out all the labels and then put it away for a couple of days and then come back a day or two later. Could you relabel it? No, not now, but someday you will be able to, I hope, because that's what will help you learn this material. But you don't have, there's multiple ways to do it. Take a slide and blank it out and relabel it. Take a slide and narrate the story, even though it's labeled and some of the story is on the slide. Take a slide, project it, and tell a friend. Teach a friend. Either in the class or not in the class, whether they're interested or not. Teach a friend the story. Kendra.
So, in the story, after protein kinase A catalytic subunits are released to do their thing, right? The next step would be that protein phosphorylation occurs and Krebs is released.
Krebs is phosphorylated and becomes activated as a transcription factor.
Okay, so Krebs is
And
phosphorylated.
aquaporin is phosphorylated and becomes inserted by exocytosis into the apical membrane.
Okay. And there's like nothing else.
Oh,
Like,
come on.
I mean, so
It's like waving a red flag in front of a bull, there's nothing else. Of course there are other things, but that's the story I want you to know. Okay?
so you're gonna ask about T-snares and, you know.
Not like this.
Oh, that's a good idea.
No!
Where are the T-snares in this story?
During
Exocytosis. T-snares, V-snares, controlled exocytosis. That's a good idea. Okay, let's talk, let's skip, I'm just trying to see where we are with time. So let's use this, well, urea is a solute. Urea it is also the excreted product of protein metabolism. You need to excrete urea because you eat protein. If you didn't eat any protein, if you had a zero protein diet, well, you'd develop a disease called kwashiorkor, but you would also not have to make urea. I'm not going to quiz you on the disease. You need to eat protein. Your body, you can produce some of your amino acids, but there are amino acids you can't produce. You have to get from your diet. And if you don't get them from your diet, you can't make some of the proteins you need to make because some of the amino acids are not there. Right? So that's why you have to eat some protein. And by eating protein, you also have to produce some nitrogenous waste, which is urea. Anyway, the point is you excrete some urea, but you also can use urea as a solute to bolster the sodium solute gradient that your loop of Henle built. So some of the solute gradient in the medulla is urea. And that's all you need to know. Urea is important as a excretory product of protein metabolism, and it's important as a solute in the peritubular space of the deep medulla. And let's not get into the molecular mechanisms. Let's just not. Unless you really want me to. Now, you might be wondering, okay, Bob, you said, let's go back. You said that if there is plenty of water, we make a gradient. But if we're dehydrated, we make more of a gradient because we express more of the sodium pumping properties of the ascending limb. The hormone that does that is called aldosterone. So vasopressin does this by the GS story, and aldosterone does this by an intracellular receptor story. Remember from self-is, there are steroid hormones, it's in there. Steroid hormones bind to cytoplasmic receptors that are usually transcription factors. It's there, trust me, I know it's there because you did pretty well in self-is. Aldosterone is a steroid hormone. Aldosterone is has an intracellular receptor when aldosterone binds to those receptors in these cells, it increases the transcription and translation of both the sodium potassium 2-chloride cotransporter and the sodium potassium pump, making more of this. So here's the vasopressin story, but in your head is the aldosterone story. Remember that the vasopressin story is acting on the collecting tubule, and the aldosterone story is acting on the ascending limb of the loop of Henle. Told you what I needed to tell you about urea. This is sort of a summary slide. Let's take a break and then do some clickers.
I'll show you.
Yeah, I'm tricking most of them. It is, like I said, it's a protein, it is a product of protein metabolism because we have to get rid of nitrogen, so it is a nitrogen-rich compound. It is colorless, and it is odorless. What are the functions of urea? You're all doing great on this one. AMP. It's not a source of energy. It is not a sodium binding molecule. So, just so you know, and you can tell your 8-year-old nephew that you learned in college what makes urine yellow, here it is. This is not important, but what makes urine yellow is urobilin, which is a breakdown product from hemoglobin. So when your body recycles red blood cells and breaks down hemoglobin, one of the products that you excrete in urine is yellow because it's a breakdown product of hemoglobin. And when we get to digestion, I'll ask you, why is bile green? It's the same answer. It is not, it has no odor. Urine has its odor from other chemicals that are excreted. So some of you may have experienced that your urine smells stronger after you eat asparagus. It's because of a thiol that is present in asparagus, and you excrete it, and that makes your urine smell. Thiols, SH compounds are usually pretty stinky. Skunks make a lot of thiols. Beta-mercaptoethanol that you experienced in is in the gel electrophoresis lab is a thiol. Stinks. Okay, so now let's talk about control. Some of this is review, and some of this is not review. So remember, we can control GFR. We talked about this. The macula densa, the smooth muscles in the afferent arteriole, the juxtaglomerular cells, all participate, and the sympathetic nerves that innervate both the smooth muscle and the juxtaglomerular cells, they all participate in the control of GFR, control of glomerular filtration. And you had a slide that was like this. So this is review. The sympathetic nervous system, norepinephrine, GQ coupled receptors, vasoconstriction. In your brain, you should be thinking calcium. Well, let's do it in order. You should be thinking phospholipase C, IP3, calcium release, calmodulin, myosin light chain kinase, cross-bridge cycling, vasoconstriction. We've done this, I've done this to you before. GQ. tubuloglomerular feedback. So if the fluid coming into the distal tubule is if there isn't very much of it. Remember, the fluid coming into the distal tubule is already hypotonic, and if it's really not, if there isn't very much of it, then there's receptors here in the macula densa that tell the juxtaglomerular cells to make renin. And now we've done this story. Renin is a hormone enzyme that cuts angiotensin from the liver into angiotensin 1. ACE, angiotensin converting enzyme from the lungs, is a hormone enzyme that cuts angiotensin 1 to angiotensin 2. Angiotensin 2 interacts with GQ coupled receptors. Da 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 da. Constriction of the afferent arteriole. Decrease GFR. Actually, complex effects on GFR, but in the case of dehydration, it decreases GFR. Okay, so that was a real quick review, but I don't want you to forget that because that's part of control of water excretion. It's not just vasopressin and aldosterone, it's this story as well. Remember, renin, angiotensin, and there's a aldosterone. That's the trigger for aldosterone came from renin angiotensin, from angiotensin. Just to be clear, where is aldosterone coming from? It's coming from the adrenal cortex. We covered this in the other AMP. Aldosterone is produced in the adrenal gland in response, and it's secreted in response to angiotensin, which has, so the adrenal also has angiotensin receptors that trigger the synthesis and release of aldosterone. Vasopressin comes from the posterior pituitary. This is not going to be on this exam because this is endocrine. Posterior pituitary is what produces vasopressin in response to dehydration. The hypothalamus pituitary axis there. Just so you know, those of you who've had AMP1 are starting to pull some of these things together. And those of you who take AMP1 next semester, probably nobody anymore. Those of you who've had AMP1 can pull some of these things together. I think this is all review. This is about reading. This is another review slide that just is cartoon version. There is the liver producing angiotensinogen. There's the kidney producing renin, the juxtaglomerular cells. There's the cut of angiotensinogen to angiotensin 1. There's the angiotensin converting enzyme converting angiotensin 1 to angiotensin 2. There's angiotensin 2 telling the adrenal to make aldosterone and turn on the sodium transporters in the ascending limb of the leukopen. Okay. So here's a flow chart of dehydration. Dehydration, low plasma volume, hypertonic fluid because you have lost water. You've sweated away water and you haven't drunk it. Low plasma volume, hypertonic. So here is the decreased delivery of fluid to the macula densa. There's renin. There's the renin angiotensin aldosterone story. Aldosterone telling the cortical collecting. If this is wrong, so cross this out. This is a mistake in your book. Plasma aldosterone telling the ascending limb of the leukopenry to increase sodium reabsorption. Making the hypertonic peritubular space. Here is the vasopressin story. dehydration, increased osmolarity are both detected by the hypothalamus. Remember, we've got chemosensors in the hypothalamus. Here we're not detecting oxygen and CO2 here. We don't detect oxygen anyway. We detected CO2, acid. In addition to acid detectors in the hypothalamus, we also have osmoreceptors in the hypothalamus. Hypothalamus tells the posterior pituitary to make vasopressin, now we're right, permeability to water in the collecting tubes. This one, the book got right. The combination of increased sodium reabsorption by the ascending limb of the loop of Henle, building the gradient, and the increased water permeability of the collecting tubule increases water reabsorption and decreases water excretion. And this is when you produce a urine that is highly concentrated. Dark yellow, lots of solute. Time to drink some water. The thirst response is actually pretty interesting, right? We could talk about the behavior of drinking. When you're thirsty, you seek water out. You go find a place to drink water. There's an interesting behavioral response to thirst as well, but we're not going to go into behavior. So let's see. Let's skip this one. Let's get to some. So here's the overview. I think I've already spelled this out. The height, sweating, decrease in volume, increase in concentration of the fluid, osmolarity. Both of these lead to vasopressin, which we have described. Both of these, well, at least the volume causes the aldosterone, which is part of the decrease in GFR story. So you don't filter it. Don't filter it. You can't lose it. Build the gradient and recover it so that we can not urinate it away, it being water. Okay, so a number of extra slides that sort of, like I said, print this slide out, white out all the words and see if you can fill it in later. Or take one of these arrows and unpack it. That's maybe a better thing to do. Why does vasopressin cause a decrease in water excretion? Tell the story that's embedded in that arrow. Why does aldosterone lead to decreased sodium excretion, increased sodium reabsorption? Unpack that arrow. What are the mechanisms that cause a decrease in GFR? Unpack that arrow. All these arrows have molecular mechanisms that I like to test you on, but they're not showing in our flow chart like this. So let's go ahead and do some clickers for the last few minutes. I haven't looked through these clickers in so long that I don't even know the answer. Okay, let's see how we do. Let's see what I once knew. I think the answer is A and C. So low blood pressure is going to decrease GFR directly. Low filtration pressure, because if low blood pressure is part of what pushes water out during filtration. And low blood pressure is part of the story here. I skipped this one, but low blood pressure, water loss is going to lead to low blood pressure, and that is going to Low blood pressure is going to do the things that we've been talking about. Sympathetic nervous system decreases filtration by constricting the afferent arteriole. If we were to constrict only the efferent arteriole, that would actually increase GFR, and aldosterone does not have an effect on GFR. Okay, which of the following participates directly or indirectly in this control of sodium reabsorption by the loop of Henle? I think the best answer here is going to be A, B, and C, and I see that many of you have it. Let's see. D, okay, D is dotted because there is a, let's forget the dotted one. That, I don't even remember what I once taught about how vasopressin can affect the sodium reabsorption. There must be some convoluted way to that. A, B, and C are good answers for this one. Okay? Everybody clear on that one? Aldosterone by the mechanism that we've been talking about for the past few minutes. Angiotensin because it increases aldosterone, and the macula densa because it makes the renin that makes angiotensin, which causes aldosterone. Which of the following helps create a concentrated urine? I think the answer is, I'm going to show them. I think the answer is A, B, and D. Let's see. Well, why not A? I mean, the proximal tubule pulls a lot of water out of urine, right? It's isotonic. So I think A is a legitimately correct answer here. The proximal tubule is important for water reabsorption, even if it's not making a concentrated tubular fluid, it's making an isotonic tubular fluid. But something more than half of the water you recover is actually in the proximal tubule. So I think A is the correct answer. The distal tubule is not a correct answer because it's what's making a hypotonic urine, not a concentrated urine. Which of the following helps create a concentrated urine? Same question. I think the answer is all, yeah. Aldosterone, as we've just been talking about, loop of Henle. Angiotensin, because it makes aldosterone, and because it constricts the afferent arteriole and decreases GFR. Norepinephrine, because it's the hormone, it's the neurotransmitter of the sympathetic nervous system that causes constriction of the afferent arteriole. Vasopressin from the posterior pituitary, collecting tubule, that way. That's it. Okay, so you've got your work cut out for you. Let's see, where are we going now?